phytosome technology, advantages and disadvantages of phytosome, application of phytosome in cancer therapy, phytosome in cancer therapy in the future, and summary. First, I will talk about introduction. Cancer is one of the most challenging diseases at present, since it cannot be complementary with current chemotherapy. Cancer is reported as one of the main reasons for death. Phytosome is an herbal extract, nanophytosome that develop from normal-sized phytosome and increasingly used in transporting drugs directly to cancer cells. In this picture, show carrier disease novel drug delivery system, which is phytosome, solid lipid nanoparticle, liposome, emulsion, and phytosome. In this study, we focusing phytosome. The delivery system is applied to herbal extracts used in cancer therapy as one technology of nanophytosome to deliver drug to the target site to cure cancer. So, nanoparticle is newly introduced patent technology developed to incorporate the standard plant extracts or water-soluble phytoconstituents in phospholipid. And next, phytosome technology is three-dimension. I will talk three-dimension. This is dimension 1, definition, 2, preparation, 3, physiochemical characterization. So, next, definition. Number 1, phospholipids are natural substances with in vivo physiological function, which make them more comprehensible for enhancing their application in designing novel drug delivery systems. Phytosomes are a type of herbal formulation with better absorption compared with that of conventional plant extract. In the picture, show difference between phytosome and ribosome. Phytosome is represents phospholipid ducts complex, but liposome represents water-soluble free ducts in cell. Comparison between phytosome and ribosome is represents phytosome associated with few molecules, mainly phospholipid and polyphenol extract, and phytosome based for oral delivery, but liposome number of molecules and even there are not connect well. So liposome prove oral bioavailability. Next, the phytosome have a better stability than liposome. This is because the former consists of chemical bond, whereas the absence in the latter. Vesicle drug delivery system that includes liposome, neosome, ethosome, aquasome, and phytosome. In the phytosome, amongst the VDDs are conicals for they have bioactive compound, and the phytosome mycel reduced nanoscope are more effective due to their invasive apparatus. So, phospholipids add to the attributes of the plant extract high market demands for products. That is, the phytosome are ideal carrier to transport the herbal bioactive compound to the target site. Next, in this picture, phytosome will go into cell membrane. This is a phytosome informed into the cell membrane. Number 2, preparation, phytosome preparation process. A. Phytosome can also be prepared simply by reversing to stoichiometric mixture phytoactive compound which phosphorylates in as a protein solvent. And next, temperature test. B. Phosphorus phytoactive compound interaction, and then evaporating is using rotary evaporator. A thin layer contains a conjugate complex of phospholipid and bioactive compound, which be formed in the bottom flask. Next, in the yeast, individual quantities of the phytosome complex that is ordinarily insoluble in polar and aprotic solvent become soluble once the phytosome complex are dry. This is in the structure difference between phytosome and liposome. This is a phytosome. The resulting suspension is concentrated by reduced pressures to active residues which can be dry and ground, natural, synthetic, and semi-synthetic. Next, in the last, the phytosome technologies. Number 3, physiochemical characterization of phytosome can be analyzed by different techniques to detect the identity of phytosomal structure, which include the following techniques: is visualization, vesicular size and zeta potential, vesicle stability, interference efficiency, transition temperature, surface tension, activity measurement, critical linearity, melting point, and spectroscopy analysis. And this is the picture text is technical analysis for physiochemical characterization of phytosome. Its use technique is SEM, TEM, AEM in microscopy, and DSC and FTR to analysis. And this is the picture is show structure difference between nanophytosome and nanoliposome. So it is nanophytosome is advantage than nanoliposome because this is advantage is phytosome have a simple production methods in comparison with other nano-delivery systems. Then phytosomes are rigorous and novel complex which have enhanced cellular uptake since they provide higher bioavailability than the using pest extracts or nanophytosome extract. And nano-delivery systems such as nanostarter lipid carrier (NLC) and solid lipid nanoparticle (SLC). And there is significant drugs, and the cellular uptake of phytosome phytoactive constitute is enhanced, but there is disadvantages associated with phytosome are adapted to the pH sensitive of phospholipid due to their zeta potential role, which should be considered in the preparation of phytosome formulation. And next, application of phytosome in cancer therapy. In this study, there is also the application of phytosome, which is application of phytosome in cancer therapy. When talking herbal extract, the herbal extract and obtaining phytosome products, which is suitable herbal delivery system. The phytosome will be developed into nanostructure. This is nanophytosome. Is a nanophytosome which is used as phytosome delivery as an anti-cancer plant diverse compound and places in a suitable herbal delivery system in the treatment of cancer cell and anti-cancer. Next, phytosome in cancer therapy challenge and future aspects. In addition, nanophytosomes are used to treat and develop future cancer therapy. In the development of phytosome, it is very useful and applied in the future. For example, in the detection of phytosome, also in detection of anti-cancer from phytosome, including the biodegradation and pharmacokinetic of MC phytosome, and the first inspection for drug delivery system and drug delivery phytosome, cosmetic phase, and biomedicals, including many clinical study at botany. So, from all the study mentioned, the development of herbal nanophytosome for the treatment of cancers can easily summarized, which is to say phytosome is a novel delivery system, which is the delivery from natural plant-based compound and cancer therapy, compound cancer therapy and products received from conventional plant extract is hydrophilic plant compound cancer therapy product obtained from conventional plant extract. This is a bioactive ability of phytoconstituents. So, nano delivery carrier is the by delivery this is by develop the system using phytosome technique by utilizing in the pharmaceutical, nutraceuticals, and cosmetic manufacture. So the main idea is to develop the delivery of nanophytosome. It's called nanophytosome delivery. That is possible and effective in treating and preventing cancer. Therefore, it can be concluded that from the reports and study that formally successfully and delivered in this way form remarkable therapeutics efficiency in animal as in humans model. In the presentation, the summary developed new anti-cancer medicine and particularly novel drug delivery system to improve pharmacodynamics and bioavailability of drugs and to deliver specific drugs concentration to cancer cells with minimal cytotoxic effects on normal cells. Thank you for attention. Does anyone have any question or suggestion? Thank you.